Now, tools of recombinant DNA technology. Now, we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools, that is, restriction enzymes, polymerase enzymes, ligases, vectors, and the host organism. So, let's try to understand some of these in detail. Restriction enzymes को देख लेते हैं. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of these added methyl groups to DNA, while the other cut DNA. The latter was called restriction endonuclease. The first restriction endonuclease, HIND2, whose functioning depended on a specific DNA molecule nucleotide. The first restriction endonuclease HIND2, whose functioning depended on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. It was found that HIND2 always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. So this specific base sequence is known as the recognition sequence for HIND2. Besides HIND2, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences. The convention for naming these enzymes is the first letter of the name comes from the genus and the second two letters come from the species of the prokaryotic cell from which they were isolated. Example, EcoR1 comes from Escherichia coli RY13. So in EcoR1, the letter R is derived from the name of E is the genus, CO means species, R means what? R means the strain, which strain of bacteria. So Roman number following the names indicating the order in which the enzymes were isolated from that strain of bacteria. The restriction enzymes belong to a large class of enzymes called nucleases. There are two kinds of these are of two kinds, like endonuclease and exonucleases. ठीक है, exonucleases remove nucleotides from the ends of DNA, whereas endonuclease makes cut at specific positions within the DNA. So each restriction endonuclease functions by inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific positions in their sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindrome nucleotide sequences in the DNA. So this is the diagram. Do you know what palindromes are? These are groups of letters that form the same words when read both forward and backward. Example, Malayalam, Malayalam, as against a word palindrome where the same word is read in both directions. The palindrome in DNA is a sequence of base pairs that reads same on the two strands when orientation of the reading is kept the same. For example, the following sequences reads the same on the two strands, that is 5' to 3' direction. This is also true if read in the 3' to 5' direction. Restriction enzymes cut the strand of DNA a little away from center of the palindrome sites, but between the same two bases on the opposite strands. This leaves single-stranded portions at the ends. There are overhanging stretches called sticky ends on each strand. These are named so because they form hydrogen bonds with their complementary cut counterparts. This stickiness of the ends facilitates the action of enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources or genomes. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and these can be joined together end to end using DNA ligases. So, this is the diagram. Now, you may have realized that normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. Separation and isolation of DNA fragment. The cutting of DNA by restriction endonucleases results in the fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode under an electric field through a medium or matrix. Nowadays, the most commonly used matrix is agarose, which is a natural polymer extracted from seaweeds. The DNA fragments separate means resolve according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. You cannot see pure DNA fragments in the visible light, and without staining, you can see bright orange colored bands of DNA in a ethidium bromide stained gel exposed to UV light. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vectors. This is the diagram.